Hello everyone, my name is Will Mosley, and this EPA P3 study covers cryptosporidium capture in sewage impacted waters. This is a collaboration between the University of Alabama and Lehigh University. The principal investigators are Dr. Mark Elliott, Dr. Kristen Jellison, and Dr. Julie Olson. There are many graduate and undergraduate collaborators. This work leverages ongoing field sampling based in rural areas of Alabama that are known to have raw sewage discharge and lab studies at Lehigh that have demonstrated both the efficacy of cryptosporidium capture on biofilms and the enhanced attachment in the presence of calcium. The objectives for Phase 1 are to identify sampling locations upstream and downstream of wastewater discharges, to design, build, and test a prototype device to increase the supply of calcium ions to enhance cryptosporidium attachment, to collect samples across a variety of field conditions and weather conditions, analyze them for oocysts and cryptosporidium species, so that we can provide preliminary evidence on source tracking and determine the efficacy of the calcium delivery devices. These activities represent the first steps to establishing what we hope will be a multi-year and multi-project collaborative research effort to explore next-generation approaches to surface water sampling. This project will incorporate people, planet, and prosperity through its research activities and community outreach. The method being used in this project enables the presence and source of the pathogen to be determined at a greatly reduced staff time and expense when compared to the standard EPA method 1623. This enables environmental justice for people through more widespread sampling in underserved areas. Across the planet, it is essential to research and develop new cryptosporidium monitoring devices so that we can reduce adverse impacts by the pathogens that are introduced into the water. Additionally, the proposed project will address prosperity through the dissemination of a less expensive and time-consuming method of cryptosporidium evaluation. We were able to conduct our first day of fieldwork in late May 2021. On this trip, we were able to take a background water sample from the creek, as well as identify the first location to place our sampling devices. Given the restrictions on fieldwork up until recently, we focused primarily on the design and testing of a prototype in situ device to feed additional calcium to the biofilm-covered glass microscope slide. You can see an illustration of the device here on the right. It's made of 2-inch PVC pipes connected by a T-joint in the middle. In this illustration, water moves from the right to the left. As it moves through the T, it impacts calcium tablets that increase the free calcium ion concentration in the water. As these tablets are eroded, more tablets fall from above with gravity. These tablets are held in the T, by a grid mesh on either side. Once the calcium ion concentration is increased by these tablets, the water continues left to impact the microscope slide embedded in the last section of pipe. This microscope slide will begin to grow biofilm and can be removed at the end of the testing period for analysis in the lab. Phase 1 focuses primarily on sampling and the analysis of samples, as well as the development and testing of the calcium tablet dissolution device. Our final 15 sampling locations will be determined through benthic biofilm sampling for cryptosporidium. And while our long-term goals for this research include substantial water quality and health outcomes, the scope is limited by the research focus and the length of Phase 1. We hope that sampling for fecal contamination can progress from grab sampling for indicator bacteria towards time-integrated sampling for pathogens, and that we can assist in that transition. Through this project, we hope to explore a more sophisticated critique of grab sampling for fecal indicator organisms and possible paths towards leveraging time-integrated sampling and emerging multiplex molecular methods to map a next-generation approach to surface water sampling. We anticipate that we will be able to identify differences between dry weather and wet weather cryptosporidium concentrations captured on the glass microscope slides, and that the slides exposed to enhanced calcium concentrations will capture more cryptosporidium oocysts than those exposed to background levels of calcium. We anticipate also that the genetic sequencing will identify cryptosporidium from both human and animal sources, and that the human sources will be more common directly downstream of known wastewater discharges. We would like to acknowledge the Auburn University Rural Studio and the Consortium for Alabama Rural Water and Wastewater, who provided important feedback and insights on the development of this project. We would also like to thank the EPA for providing the funding of this project.